Module 8, Cancer Cell Biology, Part 3. In this last part, we are going to look at some examples of tumor suppressor genes. As I mentioned in the first part, tumor suppressor genes have a normal function in cells, and their function is to inhibit cell growth and proliferation. So they're negative regulators of the cell cycle. And when you have a loss of function mutation in them, this can contribute to the neoplastic phenotype. And these mutations are often referred to as recessive because you have to lose function in both copies of your genes before you see some kind of effect on the cell cycle. Two ways in which you can lose function of these tumor suppressor genes. One is by simple mutation. You may mutate the gene such as the gene product is no longer active, or you can have something known as loss of heterozygosity, which I'm going to explain in one moment. But I need to also mention that we already talked about one tumor suppressor gene whose product is the retinoblastoma protein. And we discussed this when we were looking into the cell cycle, and this gene was actually the first tumor suppressor gene that was identified, and when it's muted, it contributes to neoplastic transformation of cells in the retina, hence the name retinoblastoma. So one way to lose function is to have a simple mutation that knocks out function. The other way is through the loss of heterozygosity. And this can happen in two ways. The first way is missegregation. So when the cell divide, we covered this in the cell cycle. When cell divide, you have appropriate segregation of chromosome to the daughter cells. And we looked at the very exquisite controls that regulate the segregation event during mitosis. So let's see here, you have a pair of tumor suppressor genes, and one of these genes is normal, shown here by the normal allele in blue, and one of those genes is mutated. So one of the pair is mutated in such a way that you have a loss of function. This cell is still okay because this normal gene and its gene product are functioning and continue to regulate the cell cycle appropriately. But sometimes during cell division, you get a missegregation event, and there you end up with an aberrant 3:1 segregation of one chromosome. This happens usually due to a defective mitotic spindle or motor control proteins. This cell on the left did not get enough of the DNA material and will die through a programmed cell death known as apoptosis. But in some cases, the cell with this extra genetic material will survive and go through its next round of division. The segregation of the extra chromosome delivery to the daughter cell is really by chance. This abnormal chromosome constitution is known as aneuploidy. This aneuploidy in cells are usually lethal to relatively undifferentiated cells that have to develop into many complex structure of an organism, but can often tolerate it in clones of cells in a tissue that have limited fates and duties. So now this daughter cell have received the chromosome in which one copy of the tumor suppressor gene is normal and one copy is mutated. This cell is still heterozygous, and so it's fine from the cell cycle regulation point of view. On the other hand, this daughter cell here was unfortunate enough to receive two mutated copies of this tumor suppressor gene. And so now this cell is homozygous for this mutant allele. It has lost its heterozygous nature of this gene, and unfortunately, this could contribute to dysregulation of cell cycle, malfunctioning DNA repair proteins or enzymes or defective apoptotic proteins that may not induce programmed cell death in a defective cell. Another way in which you can lose heterozygosity is through the mitotic recombination. And we didn't talk about this when we talked about the cell cycle, but during mitosis, you can get recombination between the homologous chromosomes, which was first discovered in fruit flies Drosophila about 95 years ago. And the molecular mechanism was described much later. Please note that this is not crossing over that happens in meiosis in germ cell lines. during development of haploid or N-numbered gametes. This is strictly a mitotic event and happens in a diploid 2N-numbered chromosome in somatic cells of an organism. So as chromosomes line up, you can get a double-strand break of homologous recombination event. And so here again, we start with the cell with this heterozygous locus where the tumor suppressor gene reside. It has one good copy and one bad copy. When you get the chromosome duplication during the cell cycle, you can get actually recombination between the homologous chromatids. So here you can see that there is an exchange of the genetic material between these chromosomes. And then during the cell division event, you get this segregation. This daughter cells here was lucky enough to get two good copies of the tumor suppressor gene. So from the cell cycle point of view, it's fine, and the cell cycle will still be regulated. But this daughter cell here received two mutated copies of the tumor suppressor gene, and it lost its heterozygosity or loss of function of that tumor suppressor gene. And that combined with the other mutations can certainly contribute to a transformed phenotype. And the same way this previous mechanism, this can lead to the loss of function of any class that is class 5, 6, or 7 tumor suppressor genes. So let's take a look at a couple of examples of tumor suppressor genes. You remember that during the discussion of the subject of the cell cycle, we talked about cyclin CDKs, and these are important dimers. They are kinases so that they phosphorylate other proteins, and they pretty much regulate the cell cycle. So there are cyclin CDKs that are found in the G1, in S, and the G2, and the mitotic phase. So an example of this is cyclin CDKs. We looked at this cyclin D CDK4. It is produced or transcribed and translated in early G1, but its function is not readily needed until mid to late G1. And so there is also an inhibitor that's present in early G1 known as P16. And that inhibitor binds to the cyclin CDK dimer and prevents it from phosphorylating proteins earlier than is needed in the cell cycle. But as the cell transitions from G1 to middle to late G1, that inhibitor becomes dissociated and this kinase becomes activated. And its role is to phosphorylate retinoblastoma or RB. You will remember that the retinoblastoma binds to that E2F transcription factor, and when retinoblastoma is phosphorylated by the cyclin CDKs, it dissociates from E2F. And then E2F can be moved to the nucleus and activate transcription of genes necessary for DNA synthesis, as well as other important cellular functions during the cell cycle. So this is all review from your cell cycle. So P16 is a tumor suppressor gene, and the retinoblastoma is a tumor suppressor gene as well. And this could be very intuitive to you. So for example, you can imagine that if you lose the function of P16, then you can never inhibit the activity of the cyclin CDK4, and they will just drive the cell cycle faster, and that will contribute to the formation of a tumor cell. You can also imagine if you lose the function of RB and cannot bind to E2F, then E2F will always be active and it will always be inducing expression of genes that are also necessary to drive the cell cycle. So two examples of tumor suppressor gene, P16 and RB. I hope it's clear to you now that how loss of function of these genes, either by mutation or by loss of heterozygosity, can be detrimental to control of the cell cycle. Another important tumor suppressor gene that we talked about already is P53. And we saw this slide previously when we were talking about the cell cycle, and this slide shows the checkpoint along the cell cycle that will halt the cell cycle if there are some problems. And there are 4 DNA damage checkpoints labeled as 4A, B, C, and D. And you can see that P53 is an important player in all 4 DNA damage checkpoints. Interestingly, p53 can arguably considered as both class 5 cell cycle control protein as well as class 7 apoptotic protein. Although it is involved in DNA damage pathway, p53 by itself is not a DNA repair protein, and so it is not considered as class 6 tumor suppressor. So here is a practical explanation. If there is a DNA damage, p53 will halt the cell cycle, which is a class 5 tumor suppressor function, and signal the DNA repair proteins to repair the DNA. If the DNA is repaired properly, the p53 will remove the block of the cell cycle and the cell can progress through it to division. However, if the DNA repair enzymes fail to repair the DNA, p53 will activate the apoptotic proteins for programmed cell death, and that is the class 7 tumor suppressor function. And we are going to talk about that in a moment. So let's take a look a little bit more closely. This is a very simple diagram about how p53 works. And the next slide is a little bit more detailed. So when DNA is damaged or mutagenized, you stabilize the p53. The p53 is a transcription factor that's normally not very stable in the cell, but upon DNA damage, it is stabilized and become a potent transcription factor. And one of the genes that it turns on, or one of the genes that it regulates transcription of, is p21-CYP. So the p21-CYP is produced. It can bind to any cyclin CDK that's around, and it inhibits the activity, and then you can get an arrest of the cell cycle. Here it is showing the arrest at G1 and G2, but you could get an arrest at any of those 4 checkpoints if there is DNA damage. So here is what this looks like in a little bit more detail. So if you have damaged DNA, this turns on the activity of a kinase known as an ATM. An ATM can actually halt the cell cycle in 3 ways. One way is that it can phosphorylate a protein known as CHEK2, which in turn can phosphorylate CDC25A and marks it for proteolysis. And this results in a very rapid G1S block, since CDC25A is necessary for the transition from G1 to S. So that's one way. The second way, and this is what we are focusing on here, is p53. So p53 is normally very unstable, but when ATM phosphorylates p53, it becomes stabilized. And when it stabilizes, it can bind the upstream region of genes and enhance their transcription. One of those genes is a p21-CYP that we talked about on the previous slide. But it can also enhance the transcription of gene whose gene products are involved in the DNA repair. If the damage is really bad, it can enhance the expression of genes that will induce apoptosis or cell death, and the cell will just die. It can also sustain cell cycle arrest until the damage can be either corrected, or if it can be corrected, then the cell will die. The third way in which this ATM, and this is related to p53, can halt the cell cycle upon damaged DNA is that it phosphorylates a protein known as MDM2. The normal function of the MDM2 when it is not phosphorylated, it binds to p53 and mark it for its degradation or proteasomal degradation. So this is why p53 is unstable. It's because this MDM2 protein binds to p53. But when MDM2 is phosphorylated, it cannot interact with the p53. And so this contributes to the newly acquired stability of p53. You have this increased pool of stable p53. I need to mention one other thing about MDM2. In transformed cells, there is a protein known as p14-ARF that's often expressed, which produces abnormally high levels of p14-ARF, and it combines to MDM2 and prevents the interaction of p53. That inhibition of the interaction of MDM2, again, that increases the pool of stable p53 in the cell. So I also need to mention to you that MDM2 is often affected in tumor cells, and it is hyperphosphorylated in tumor cells. So all these could lead to considered as loss of function in various ways that may lead to neoplastic phenotypes. All right, so in more than 70% of the cancer, p53 function is lost. That's why p53 is often called as the guardian of the genome. The p53 is mutated in such a way that it cannot behave as a transcription factor. So it cannot turn on expression of genes involved in DNA repair, cell cycle arrest such as p21-CYP, or even genes that are going to induce cell death if the damage is substantial. And this diagram shows the frequency of the mutations in certain parts of p53. So this is the p53 here. This is the N-terminus, and this is the C-terminus. It has this 1, 2, 3, 4, and 5 DNA binding domains here in the purple shading. So when this protein is folded, these are the regions of the protein that actually interact with the DNA and turn on transcription. And what you have along here are these bars which indicated the frequency of mutations. So this is at the amino acid level, and you can see that mutation in p53 at this level. So these are relative levels of the mutations, and you can see that there are hotspots. Like for example, this amino acid like R175, arginine 175. This is mutated in high frequency in cancers. This R248 at position R249, this G245, glycine 245, R273 or R282. You can see that these are the mutated very frequently, and it's no surprise that these hotspots, these mutation hotspots, are in DNA binding domain in such a way that p53 can no longer interact with the DNA. It can't turn on genes, so that it's a loss of function. This is a classic example of a class 6 tumor suppressor genes that are known as BRCA1 or BRCA2 and often known as BRCA1 or BRCA2. They are implicated in hereditary breast cancer, but also implicated in ovarian, prostate, pancreatic cancer, and melanoma. These mutations are inherited, and therefore a genetic diagnosis is available. If an individual has certain BRCA1 or BRCA2 mutations, by the age of 70, they have more than 50% chances of developing some form of cancer. So how these mutated genes cause cancer? The BRCA1 or BRCA2 genes products repair double-stranded DNA breaks involved in homologous recombination, where the homologous chromosome is providing the template. So an impaired or loss of function of these DNA repair proteins can lead to gross chromosomal loss, rearrangement, or translocation. We have seen this similar example of this in the Burkitt's lymphoma in part 2 of the topic. This gross change can lead to altered transcriptional regulation, protein ubiquitination of the cell cycle control proteins, leading to cancer. So let's end by mentioning the current treatments and some future prospects. Right now, for certain cancers, surgery is often a part of the treatment, if not the only treatment. It is very effective for primary benign tumors. It is less effective for metastatic tumors. Radiation is often used for some tumors, which effectively kills tumors, but also kills normal cell. So this is the drawback. Chemotherapy, many chemotherapeutic agents are designed to, not all of them, but some of them are designed to interfere with the mitotic apparatus. So as these cancer cells are rapidly dividing cells, they require a functional mitotic apparatus. But if you disrupt the mitotic apparatus, you can prevent the cell division in these cancer cells. But if you can disrupt that mitotic apparatus, then you can prevent the cell division in these cancer cells. So a lot of chemotherapeutic agents target the mitotic apparatus. Another type of chemotherapeutic agent is actually a DNA damaging agent. There's a lot of chemotherapy which damages the DNA in hopes that if you damage the DNA, then p53 will kick in. And if you damage the DNA sufficiently and p53 kicks in, those cells will die. They will go through apoptosis. The problem is that in more than half of the cancer cells, p53 is mutated, and so the cell cycle is not arrested. The cells do not undergo cell death in response to DNA damage. The other drawback to these family of chemotherapeutic agents is that the normal, fast-growing cells are also affected. And that's why individuals who are going through chemotherapy often lose their hair and have side effects associated with the skin and intestinal tract because these are very fast-growing cells. So what is the future of cancer research? Listed here is just a few of the possibilities. You don't need to worry about this for the test or the exam or the quiz. I just want you to be aware of what the current research is. So you may have heard about precision medicine, also known as personalized or individualized medicine, and this is the hope that in the future, all people will be treated based on their genetics and history. This is a lofty goal, but there has been some progress. Some cancer patients have already benefited from targeted therapies. Immunotherapy, which is used of a patient's own immune system to fight their cancer. Immunotherapy is becoming more effective day by day. For example, antibodies to the class II oncogenes, such as receptor tyrosine kinases, that when mutated are involved in or associated with cancer. Generating antibodies that bind to them and block signaling from them has been very successful in the treatment of breast cancer. A drug called Herceptin, which targets the HER2 receptor, is very effective at stopping the signaling from the HER2 oncoprotein. Two new immunotherapies are one immunotherapy checkpoint inhibitors, not to be confused with the cell cycle checkpoints, and two is CAR therapy. The immune cells, the T cells, have an off switch to keep the immune system in check. There are molecules on the surface of the normal and healthy cells that tells the T cells to leave it alone. Cancer cells sometimes find ways to use these molecules or immune checkpoints to avoid being attacked by the immune system. Several drugs have recently been developed that target these immune checkpoints and restore immune system function. This therapy has shown very promising result, eliminating cancer completely in some patients with melanoma. The CAR therapy or chimeric antigen receptor therapy is a strategy by which the patient's T cells are collected, sent to a lab to be genetically engineered to attack cancer cell surface protein, and then infused back into the patients. Although there are severe side effects with this therapy, the outcome has been surprisingly successful for treating advanced blood cancers. Vaccines. Some cancers are caused by viruses such as human papillomavirus, HPV, which is linked to cervical cancer. Development of vaccines for various cancer continues. Epigenetic drug. Some genes are influenced by the environment, and new drugs are being developed to target epigenetic enzymes that regulate the cell's genetic programming. The idea is to return the cancer cells back to their normal state rather than destroying them. Metastatic drugs. So metastasis is the spread of the cancer. Developing drugs to control the metastasis has been an ongoing challenge for many years, primarily due to the difficulty in isolating the metastatic cancer cells. But in recent years, the scientists have identified some important genes that drive the spread of the cancer to other organs. So promising new treatments to control metastasis are on the way. A whole new frontier of cancer therapy is being developed with several ongoing clinical trials using chimeric viruses. These chimeric viruses target the cancer tissues either through cancer cell-specific receptors or by using the property of excessive angiogenesis. Excessive angiogenesis means that the malignant tumors have way more blood vasculature and increased blood supply to cater their rapid growth. These oncolytic viruses either destroy the tumor by their cytopathic effect or cell-killing ability, or these viruses can be used as a vector and loaded up with a drug or toxin to supply to the malignant tumors to kill them. Eventually, the virus is cleared by the individual's own immune system without any harm or minimal toxicity to other tissues. The last one is the gene editing technology. For years, there has been substantial effort to replace the p53 or p16 in tumor cells. One of the challenges was developing a technique to deliver functional copies of p53 or p16 just to tumor cells without affecting cell cycle regulation in normal cells. But now there is a new technique for gene editing, CRISPR, which is clustered regularly interspaced short palindromic repeats, which is a very exciting breakthrough which allows scientists to make precision edits to any DNA. So now scientists potentially have the ability to replace the faulty genes with the normal genes and new ways to edit genes as well as treat cancer. And so I hope that you can appreciate that neoplastic transformation of a cell is really when the cell breaks the rules. There are exquisite controls on the cell cycle, and when those rules are broken or when those regulatory events become non-functional, you get cancer. So I now hope you can appreciate that from a cell biology standpoint. This concludes Module 8, Part 3 of Cancer Cell Biology, and also concludes Module 8.